feeling the breeze coming out from Teddy's room. His window was open this much and the heat was on 70. Well,
You ready, Dr. Mascala?
I am, yes. Let's start.
Okay, perfect.
All right, good morning. We have quite a nice number so far, and hopefully more. All right, to all the students, good morning. I hope that you got some rest after the biochemistry exam. And we start now with this review today on, okay. Okay, so here we are. Of course, these are all, this is, what do we call this? A smear, right? And what do you think this could possibly be? Look at these red granules. You can see even a low magnification. So how would you call that? Anybody?
That's a granulocyte.
This is a granulocyte, and we can be a little bit more specific because the granules can be even seen at this low magnification and very, very red. So
it's an eosinophil.
Eosinophil, perfect. Okay, wonderful. And so here we have a row of what? These are granules, but they are not that red, right? All right, so neutrophil. These are neutrophils. These are neutrophils, and this is the youngest of the three. This is the oldest of the three. All right, so it's nice to see, because this still has a little bit of a band, and now it's starting globulation. Here is starting globulation. All right, so, and here, obviously, this is now, yes, we can see all these granules that are very, very conspicuous, right? Sometimes it looks like the eosinophil, instead of being bilobed, may have a third lobe. So nothing wrong with that. It's still a normal eosinophil. And what is this row here?
Basophils.
Basophils, how can we tell?
Dark staining.
Because I'm sorry, could you say that?
Dark staining, the granules are dark.
Yes, very, very conspicuous. All right, so we can see, in fact, this is the younger of the three. And here we can see a little bit of the nucleus, a little bit. When we go to a fully matured cell, not that this one is not matured, they are all active in their function, but this is the oldest of the two, and the nucleus is masked by the conspicuity of the size of the granules. All right, so here we have this row here. What do you think is represented by? What are these cells?
Lymphocyte.
Yes, how can you tell?
Because they're round and they don't have as much of an indentation as a monocyte would.
Yes, there is a very small indentation if they show it. In fact, this one doesn't, and this one doesn't either. But what they have is the cytoplasm. It's always the ratio of cytoplasmic to nucleus is the cytoplasm is much less comparing to the other cells that we see here. Yes, now you can argue that the cytoplasmic is very little too in this cell, which we call what? This is a monocyte. A monocyte, perfect. And these are younger monocytes. They're beginning to have, this one's beginning to sort of making this indentation. This is a little more, this is sort of a more mature monocyte. All right, so here is a smear of a, what could this possible a labeled empty, almost structures are.
Adipose tissue.
This is adipose tissue, and there are a lot of these. This do remind you of blood cells? I would say yes, right? But they are in many different shapes because this is a bone marrow smear, all right? And we gave all these cells. In fact, these are, oh look, I even left the label here. These are erythrocytes. These are mature erythrocytes, or at the best, reticulocytes. They are ready to go. And this is pretty big. What do you think that could possibly be?
A megakaryocyte.
Yes. And so a megakaryocyte, is there any relationship between this megakaryocyte and what you see in these figures here?
They produce the platelets.
That's right. And here these are the 1, 2, 3. Well, it's the same here. This is now a regular smear, very high magnified. All right, and this is 1, 2, 3, 1, 2, 3.
They're platelets.
They're platelets which came out of this large megakaryocyte. All right, and this, it's definitely the largest cell in the bone marrow. Okay, so these are formed elements because they don't have a nucleus. And so the red blood cells too are also called formed elements. I don't know whether that was said in the lecture. If not, don't worry. Okay, so here we are. What could this possibly be?
Plasma cell?
The plasma cells are never in circulation. The plasma cells are only found in connective tissue.
Lymphocyte.
It's a lymphocyte.
Lymphocyte, sorry.
Yeah, that's probably what you meant. Yes, thank you. Okay, so this is a lymphocyte and it's a small lymphocyte. All right, this is really virtually almost no cytoplasm. All right, so here we have something, and this 1, 2 are, of course, we discussed those. These are eosinophils. All right, here are 3 granules. See how big they are. And then we go to the basophils, which have even larger granules. The smallest granules are found in the neutrophils. And what is this one?
Bar body
Bar body. And what is the bar body?
in the neutrophil.
In the neutrophil. And what is it? What does it tell us?
It's a
an
chromosomal
inactive
condensation.
Yes, it's one of the 2 X chromosomal condensation, all right? And that means that this sample, at least this one, all right, was taken from a female, all right? Okay, so here, again, we have here, now we know, right? This is quick now, all right? Because we are reviewing, and we don't want to waste the time though. So this is a, what is this?
Monocyte.
Okay, this is a,
Lymphocyte.
this is a lymphocyte because of the cytoplasm. And see, there are some exceptions. This is a monocyte because there is a lot of more cytoplasm, even though there is no indentation. All right, so, but it's okay. You are going to have perfect pictures. Oh, I'm sorry about that. Okay, so this is a,
Monocyte.
yes, par excellence, perfect. This is the perfect one. And all this here, this is a, this and this,
Basophil.
the same, right? Yes, and this one is also a basophil. This is the nucleus we can even see. All right, now we get here. And these are naturally erythrocytes, right? Yes, how they are, why they have this whitish area here? Because they
The
are
nucleus has been extruded, yeah.
yes, and because they became biconcave, right? All right, and there is a reason for that, because that allows for a more efficient exchange, all right, and also because of the skeletal, the cytoskeleton, because of this conformation, they can move around better when they have to go through a capillary. All right, so these are dumbbell shaped as they seen in this electromicrograph. All right, and here is a platelet. And this here, what are they? What is this R stands for?
Reticulocyte.
Reticulocyte, yes, they are just about or just came out from they just left the
marrow.
Bone marrow, correct, okay? And what is this, what are the structures here? What is this bluish basophilic stain?
Is it
Ribosomes.
These are some ribosomes that are still there. All right. Okay. So do we see this in circulation very often? If we do a blood smear, for example, we expect to see them?
No.
No, rest. But what happens if they are in the blood smear?
Indicates leukemia or some other blood cancer.
It's anemia.
Yeah.
It's anemia. All right. It means that there is a lot of erythrocytes that are metabolized and destroyed in the spleen. And so then there is a demand for very, very much and very fast demand of reticulocytes. So it's anemia. All right. And so how do we treat severe cases of anemia? How do we treat that? There is something that can help, right?
You give them erythropoietin?
Erythropoietin, all right? That is desynthesized in the liver. Okay, so now, what? Okay. Okay, so here we have what? This is easy. I mean, right? We know this. Even before the lecture, we would know this. All right? This is a section of. Look, I have this eosinophilic staining structure here. This looks like a nucleus, doesn't it?
It's skeletal muscle.
And it's a skeletal muscle, correct. It's a skeletal muscle, all right? It happens to see a muscle spindle here, all right? So it's just, we're not going to spend much time on that. But we are going to spend much time on this section. Not much, as much as it's needed, right? And so we have this connective tissue here, right? There is nothing outside here. Really nothing all along. So how do we call this connective tissue in skeletal muscle? This particular one, which is outside the muscle. What
Is it epimysium?
Epimysium, very good. Epimysium, all right? So the epimysium extends in between groups of muscle fibers, all right? These are muscle fibers, okay? And this now becomes what? All this here, all
The
this.
perimysium.
Perimysium, it's around the fascicle, all right? But then we have these fibers here, skeletal muscle fibers, all right? And this skeletal muscle now this muscle is cut in a transverse section. And so that we can see the connective tissue better around each muscle fiber, right? All these are muscle fibers. And so how we call that?
Endomysium?
Endo means inside, all right? Is inside the fascicle. And it's very, very close to the muscle fiber. What do you think this is? And it's related in any way to this one.
Is that muscular dystrophy?
Yes, this is Duchenne muscular dystrophy. All right. And this Duchenne muscular dystrophy, how does it come about? It's a recessive condition, all right? And where does it start? Who would say be the culprit culprit? You know, who is responsible for this condition?
Dystrophin.
Actually, dystrophin, all right? So dystrophin, what does it do? Dystrophin attaches to a system, all right? We don't need to know. It's the dystroglycan complex, which attaches to the membrane, all right? To the plasma, to the sarcolemma. And so if there is a mutation in dystrophin, what does that happen? What does that mean? What does dystrophin is connected to the fibers? So if dystrophin is it's mutated, cannot do the job that it has to do, right? And so these fibers are not able to do the job that they have to do, contraction. And so there is going to be not only they're not able to, but also because of the disruption in the plasma membrane, there may be calcium coming in. And so that is involved in degeneration of the muscle fibers, all right? And so we have all this infiltration of small fibers that, because there is an attemptive regeneration, but is not possible because the dystrophin is not there to make a fiber to be normal as this one. Am I making sense? So basically that's what happens. And that's what we have infiltration then of connective tissue, because if there is no skeletal muscle, connective tissue, fibroblasts are going to proliferate. They try to do something, but it never reaches the normal condition of the muscle. And so that's how it appears. So these are regenerating fibers, and we have these fibers that are trying to do the job and they become hypertrophic. All right, with that said, all right, when Duchenne muscular dystrophy appears in life, very early, in very young people, all right, the early symptoms are seen at very early stage in life, all right? And so we have then this degeneration. We discussed all the points that actually lead to muscular dystrophy, Duchenne muscular dystrophy. Okay, now here, what do we have? What do you think this is from? Okay, let's take this one to this one. If I put a bar here, what would this be? What would you call this?
Myofibril.
Myofibril. Okay, so a skeletal muscle fiber has a nucleus that is peripherally located. All right, here is another one. A skeletal muscle fiber has a nucleus that is, not many nuclei actually, that is peripherally located. Therefore, this is a muscle fiber, muscle fiber, muscle fiber. All right, now we have, this is a
sarcomere.
What is that? This one. From here to here.
The A band.
The A band is the dark band. All right. And what is this? This is the dark band. the A band. And so here we have this one here is 1 and 2. On other side of the dark band, we have the light band, also called the I band. So here we have I band, okay? From here to here is what? This is very high magnified. I'm even showing it to you, all right? So this is
The length of a sarcomere.
yes, this is the sarcomere, which is flanked by the Z discs. All right, are you with me? All of you? And so therefore, what do we have? What are these thin, very almost invisible structures, even at this high magnification? What do we call them in the I band or light band?
Actin.
The Z.
These
Okay,
are
yeah.
actin filaments, all right? And what do we call these bands right here?
Myosin.
Exactly, these are the thick filaments or myosin, perfect. And so what happens during contraction? For contraction to occur, which of the two filaments is going to move?
Actin.
Exactly. These actin filaments are going to move and go more and more inside. All right? Okay. And so here, what do we have? I like it to see. Let's see if we can make so this here is high magnification of this fiber, 1 and 2. You think? This is a myofiber, a low electromicrograph, which would be then this myofiber here, right? Because we said that this is a myofiber. The nucleus is located peripherally, all right? Okay, so this one and this one, are they called the same? Yes. This is light microscopy. This is low EM. And here we made this because we want to see the myofibrils. These are the myofibrils. The myofibrils are made by sarcomere aligned in tandem. 1, 2, 3, 4, 5, and 6 for this segment of myofiber. There's a lot of connective tissue here. And this connective tissue here is the connective tissue which we see here, which is the endomysium. Are you with me? Yes? Okay, so this is a myofiber. Myofiber. If we want to see the myofibrils, we have to go to at least low scan electromicrograph. This is a myofibril. Myofibril 1 and 2. And what's in between here? This is myofibril. Oh, we have mitochondria. We don't see them. We need a higher mag. But here we can see a little bit because now we have one sarcomere. And this sarcomere is highly magnified. It's just one of this, like this one, which is magnified. And now we can see glycogen, and we can see the myofilaments, etc., etc. We see a mitochondria here, right here. All right? Okay. And are we okay with that? Yes? I
Dr.
see you
Mascella,
one number. Yes.
can you just point again to where the sarcomere is in the lower right picture? I just want to make sure.
I'm sorry, D? Can I point it
Yeah.
to?
Yeah, can you point to where the sarcomere is in that lower right picture?
This is the sarcomere. This one, right? Oh, you mean on this one? It's this one.
No, you were right. I got it. Thank you.
Oh, okay.
That one.
Good. I'm so happy to hear that. Okay. All right. A little bit more. What is this? I'm not going to say anything now. What is it from here to here? You are going to teach me now. Come on.
The
Sarcomere.
Sarcomere. And that is the functional unit of the skeletal muscle, correct? Okay, because that's how we have contraction. When sarcomeres contract, then we can move. So sarcomere is the functional unit. Okay, now here we have a little bit of something. This is a T, which stands for transverse. What is this?
It's a T-tubule.
Transverse tubule, perfect. A T-tubule. And what is this T-tubule in between?
Cisternae.
Terminal cistern, right? Of the sarcoplasmic reticulum. See, this sarcoplasmic reticulum, it goes all the way down, dilates, and makes this terminal cistern. This is this one here, all right? Okay, so that's so what happens? I have a motor and the plate here, all right? So these are junctional folds. Somewhere here, this is a Schwann cell. Here is the nerve, all right? The nerve comes with an action potential. All right, the action potential then acts so that acetylcholine is released. Acetylcholine gets in here, and it's going to attach to receptors. The receptors are going to change the membrane, and with further, which we are not going to talk here, events, we are going to elicit an action potential. Okay, so now here somewhere in this scheme of fibers, there is a motor in the plate. All right, then the action potential of the fiber is going to be taken to the T-tubule, because see, here is the membrane, here is the membrane. So here is the membrane. The action potential runs here, all right? So forgive me. So this is the fiber. Here is the membrane, invagination, T-tubule, and these are the cistern, terminal cistern. And here we have, then with the action potential, we're going to have calcium channels, which are going to be opened upon the arrival of the action potential. And they're going to release calcium, and they're going to bathe all these myofibrils. All right? And so if calcium is around, then there is muscle contraction. Okay, so here now we have another tissue. All right, so this is from where? This is from?
Cardiac tissue,
Yes, what are these?
intercalated discs.
Intercalated discs. And what is this?
Purkinje fiber. Or no?
Also intercalated discs.
Yes,
Yeah,
very good, very good, very good. Yes, this is, they are made of one transverse component, all right, which runs transversely to the length of a cardiomyocyte, all right? And remember, cardiomyocytes are branching, all right, and also diverging. Okay, so this is the transverse component. What is this junctional complex here on the transverse component?
Fascia adherence.
Yes, it's even there, fascia adherence. All right. Now we have also a longitudinal component, which is this one here. What's on the longitudinal component? This is one, and this is a little bit more deviated, but this is another junctional complex. All right. This component says this is my junctional complex. All right. The longitudinal says to the transverse, don't take it from me. All right. This is what type of junctional complex? It's a desmosome.
Gap junction.
No, a desmosome. Whenever you see this intensity, electron dense structures, it's either the fascia adherens, which is much larger, all right, or the desmosome, which is much smaller. So here is a desmosome, here is a desmosome. Now the gap junction is this one. The gap junction doesn't need to have this electron dense, this structure here, all right? So this is the gap junction. And so the gap junction are on the longitudinal component. Why do you think they are on the longitudinal component? Because is at the longitudinal component, the current from the gap junction is going to be released or expanded or come in contact with this, what is this one here now? This is another, on this side we have a Z line. This is a portion of a sarcomere, all right? And so it's via this gap junction, the current is going to expand and around the sarcomere, all right? And so why we have this? We have this because the contraction of these fibers is synchronous, all right? And so when we have this current given at the same, provided at the same time, then the heart contracts, okay? By the way, where is the contraction of the heart initiated?
Say node.
A say node, which is the sinoatrial node, all right? Okay, all right, are we okay with this? And these are the T tubules too, all right? Okay, so, and this is a sarcoplasmic reticulum, all right? Let's go on. All right, so this is, again, this is what?
The intercalated disc.
And so we are in the heart, all right? Thank you. And so this is now the skeletal muscle, all right? So here is almost a triad, all right? But this is definitely a triad right here, all right? So there are differences, and that is the T tubules in the skeletal muscle lay in the eye, at the junction of the eye and a band. But in skeletal muscle, they run at the level of the Z lines. All right, here is a Z line. All right, here is a Z line. All right, one thing that I want you to see is that here there are a lot of mitochondria as opposed to the skeletal muscle. Why? Because the heart contracts continuously, all right? It's not under under our will. And so we need a lot of energy. What is this? All this now. All this. Or there's any difference between here, here, and here, here, and here? So this is a smooth muscle. This is a smooth muscle that is cut in transverse section. This is
Cardiac muscle.
point, exactly, branching point, and this is an intercalated disc, and this is another branching point. So this is the cardiac muscle cut in a transverse section, and this is branching point when, as they see, in longitudinal section. All right, now what do we have here? We have a lot of branching point here, centrally located nuclei. All right, centrally located nuclei even in smooth muscle. But here we have centrally located nuclei, but we see these dots. These are the myofibrils that are in the heart, all right? And here we have the myofibrils, all right here. And here we have myofibrils, all right? Here the myofibrils are not seen very well, all right? But these are the typical cardiac myocytes, right? Yes, yes, with centrally located nucleus, all right? These are the myofibrils. It's a little bit of glycogen in them because these fibers need energy, all right? And it's at the expenses of glycogen. And we have these fibers here. Some of these fibers are also showing a little bit of myofibrils right here. But they have this very large empty looking space. Here is the nucleus. And this space is around the nucleus. What could these cells are? Or fibers? Here are some of the myofibrils, because here we have an idea of the sarcomeres. So what is this occupied by? A lot of glycogen. Are you with me? What are the fibers that have a lot of glycogen? Also, there is a lot of space here because this is beneath the endocardium. What's beneath the endocardium? There are cardiomyocytes, but they are specialized, so to speak, cardiomyocytes. They are involved with the signal that comes from the sinoatrial node, which goes into the bundle of Kiss and ends in these myofibers, which are called Purkinje fibers. All right, Purkinje fibers. They're also called the cells, but they are real. They make fibers because they make a fiber that continues with the bundle of Kiss. All right, okay. And so here we have the heart. This is the here is the outside of the heart. This is the mesothelium. All this is epicardium. There is a lot of fat, but then we have the myocardium. And then here we are going to have the endocardium. All right. Now, let me ask you, the endocardium is corresponding to what tunica in the vessels? Because remember, the endocardium is in the, it's aligns the lumen of the ventricles and the atria?
Intima.
Exactly, tunica intima, perfect, very good. All right, and so endocardium, and we have myocardium. Why is it called the myocardium? Myo is a prefix of muscle, right? So this is the myocardium. It's the muscular portion of the heart, of the wall of the heart. And here we have the epicardium. Epi is around the heart, endo is inside the heart. Are you with me? Okay. And here we have some fibers. They are in. So here is the endocardium. And this is the subendocardium. And that's where we find the Purkinje fibers. Because the bundle of keys, so here is the inside of the chamber of the heart, and here is the endocardium. And so this portion here is shown to see how the fibers run under the endocardium. Okay. Is that clear? Yes? Okay. Am I speaking slow enough so you can take notes?
Dr. Michelle,
Yes.
what was the top left picture in that last slide? Did you go over that?
This one?
No, the blue one.
Oh, this one.
The top
This
left.
is the endocardium. No, which one? The blue?
Yeah.
Oh, the blue. Okay. All right. So these fibers here, this is muscle, right? This is muscle. This is a muscle trichrome. Staining, you don't need to know that. But with this stain, which is different than H&E, which is this one, it stains the collagen blue, a red or reddish, the muscle. Are you with me? Okay. So what's showing here is the regular, so to speak, muscle cardiomyocyte stain very, very red because there is not as much glycogen, okay, as there are in these Purkinje fibers here. Are you with me?
Yeah.
Thank you for your question. I did not go over it. Is it clear for everyone? Yes? Okay. And so these are the nuclei of the Purkinje fibers. All these are Purkinje fibers. So if these are beneath the endocardium, what would be here if it were not washed away? Blood, because this is one of, most likely, one of the cavity of one of the ventricles, I should say. All right. So there's nothing here, and here are endothelial cells. All right. And so, and here is the equivalent of the intima of the blood vessels. And here is, so this is all this endocardium, and together they make this region. And here is the subendocardial region where the peripheral, where the Purkinje fibers are found. Any questions on that? No? Nobody has questions. Okay, let's move here. Okay, so what do you think? What do you think this? Remember this from the lecture? This is a schematic view of a smooth muscle fiber. All right. Here is a portion seen in the electromicrograph of a smooth muscle fiber right here. Right. And what we have here is that here is the outside, right here, of this particular fiber. Here is the nucleus. These are mitochondria. This is endoplasmic reticulum, all right? And here are filaments, all right? And here are the it will come to me now. These are the dense bodies, which are the equivalent of Z lines in the muscle, all right? And here we have ribosomes, all right? Here are invagination of the membrane of this particular fiber, smooth muscle fiber. The C stands for caveola or caveole in this case when there are two. What are the caveole for? They are for some of the messengers, like an hormone that has, well, here is a receptor, but other messengers do not have a receptor or have a receptor in the caveole. And through this caveole, we have the entrance in the muscle fiber. All right? And so the hormone receptor is here, all right, on the caveole. On the hormone receptor is on the caveole. And that hormone receptor or another substance that is stimulatory for contraction of smooth muscle enter through this. They are here, there are receptor here, and then it's carried inside, and this is called the second messengers. All right? Okay, so caveole are located on or invagination of the smooth muscle membrane, and they are the way whereby second messengers enter the smooth muscle. What happens after that? All right, there is release of calcium from the smooth endoplasmic reticulum. All right, and the calcium is going to bind to calmodulin. We make this complex. This complex allows for activation of this enzyme, and we go from inactive myosin to active myosin. Okay, that's in summary. All right. Okay, so what also do but remember, caveole are crucial in most cases, all cases, or all cases basically, for muscle contraction. Okay, and okay, so here we have, see, here are smooth muscles. All right, all these are smooth muscles. Okay, and so on each of these, let me see. So here, let's take one fiber right here. The membrane is equipped with those caveole. This is smooth muscle in a vessel, and these are in different organs of the body, because that's where smooth muscle is found, right? It's the muscle of the hollow organs. All right, now we move to another topic, and that is, this is the, what could this possibly be? This, right
Tunica
here.
media, elastic artery.
These are elastic fibers, all right? And it's tunica media, and it's very large, all occupied or all containing all these elastic fibers. We also have muscles right here, all right? So this is the intima media, and this is the adventitia, all right? Okay, so this could be What do you think this is then? If you have to give a name, anatomically speaking. How do you call this vessel with all these elastic fibers? The aorta. Isn't it the one? I mean, so far you have learned that this is the one that contains most of the elastic fibers, because this branch of the aorta has two branches, two iliac branches, all right? Then in this branch, we begin to have more muscle and less this wiggling, these undulating structures or elastic fibers. Are you with me? All right. These are the elastic fibers here, all right? There are not many. Right here we have muscle. This is all muscle, all right? A little bit of, this is elastic fibers. This is what? What is it called, this one here?
Inner elastic membrane.
Yes, which we don't see in the aorta very well, because we have all those fibers. We begin to see that when we begin to have muscle in the wall of the artery. And therefore, when this iliac arteries is going to give rise to this artery, which we call muscular, which are also distributing arteries, all right? What these arteries are called now? I just told you, muscular, right? Okay? We don't have basically elastic fibers in the media, correct? But we do still have this right here, all right? Internal elastic lamina. So conspicuous that we don't even see where is this nucleo from? Where is it contained? In what cell? All right, what are these cells here? This nucleus here.
Endothelium.
The endothelium, correct. Endothelium, all right? These are the endothelium, all right? So the endothelial cells, beside the lining, the lumen, what else do they do? And what happens here in some non-normal conditions? Let's say that we have a lot of LDL in this lumen.
Things like atherosclerosis and arteriosclerosis.
Yeah, so what we have is that, unfortunately, these endothelial cells, poor ones, are going to be injured, all right? And so with injury of the endothelial cells, then we have a cascade of events that leads to, most likely, arteriosclerosis, right? You know that, all right? You know that, you know that. You probably have heard of that too. Okay, so the endothelial cells are safeguard for the wall of the arteries. Okay, too, right? Okay, so here we know what it is. All right, what is this here? What is this from here to here? From here to here. What do you think this is?
An elastic artery.
Which one? Elastic?
Elastic.
Did you see elastic or muscular?
I said elastic.
No, it's not.
Oh yeah, I see the muscular
These are
layer.
not elastic fibers. All right, these are elastic fibers here. And outside here, we also have elastic fibers. But here, we don't have elastic fibers. This is the tunica intima of vein, right here. Okay, let's
So it's a muscular artery?
Muscular artery, all right? Okay, so this is a muscular artery. And the muscular artery has an external elastic lamina too. And there are some elastic fibers even in the adventitia, very short adventitia of the elastic fibers of the muscular artery. All right. So this is the internal elastic lamina. Here we have the endothelial cells not seen at this magnification. This is the tunica intima. All right. And here we have the external elastic lamina, which is continuous. And it's this one here, seen at higher magnification. And then there are some elastic fibers in the adventitia. Okay, what are these right here? What do we call? I'm sorry, what?
Vasa vasorum.
Vasa vasorum, all right? Yes, so these are, here looks like a venule, post-venule, post-capillary venule, and this is a larger one in the vasa vasorum. And, oh, and I have a question for you. So we discussed the tunica, the equivalent of the tunica intima in the cart, which is the endocardium. We also discussed the myocardium, which is equivalent to the tunica adventitia of the arteries. And what is the equivalent of the tunica adventitia of the cart? That is the epicardium. All right. And what is the equivalent of the vasa vasorum in the cart? What makes the vasa vasorum in the cart?
Coronary arteries.
Coronary arteries. They run exterior to the cart. And then once they eventually are going to give branches, which are still outside the cart, but then when they're very small, they penetrate the cart, okay? All right, great. Okay, so what do we have here? All right, so here we have, this is a tunica media, all right? And here is also a tunica media. This is smooth muscle, right? We showed before so much smooth muscle, all right? Smooth muscle and smooth muscle. So this is an art to see all these undulations here, this sort of sinusoidal aspect of the tunica intima, right? Okay. They're not very obvious here, only a little bit. So what is the difference between this vessel and this one?
Artery
Fewer
versus.
layers of smooth muscle.
Okay, what is the inne? What is the inne? What vessel is inne? That same muscular artery, all right? Okay, so this is the adventitia that is around the tunica intima, all right? And then we have, and this one, what do you think it is? What do you think this is?
A vein.
A vein, very good, all right? So what is the difference between the arteries and the vein? All right, they have, the muscular artery has a thick tunica media and a very thin, considerably comparing to this, tunica adventitia. Or the tunica intima is double the adventitia, at least. All right, here instead in the vein, we have the tunica adventitia, which is almost double the thickness of the tunica media. All right, okay. So what do we have here? These are endothelial cells, smooth muscle, and here would be the tunica adventitia. So this is muscular artery. And here, no, this is the vein. This is the only vein I have. But remember, the vein has the tunica media of the vein is half of the tunica adventitia. In fact, we can see even here and even here. So this is a vein because this is the tunica adventitia. Here they are meeting almost, they're very close to each other, closer to each other, artery and vein, so there is less adventitia. So we have to consider this, all right? So this is the adventitia, all right? Double the thickness in the vein. And this is the adventitia, but here, all right, of the artery. Okay, is that clear? Is that clear? I see a few faces that are perplexed. Are there questions? I see some perplexed faces. You can ask me questions.
Stella?
Yes.
Dr. Stella?
Yeah.
Hi. So in the lower left corner, that would be a vein, right?
Which one? Here? Here with this one? This is a vein.
Yes. Okay.
And this is an artery. All right? Is that clear?
Thank you. Yes.
A thick, very thick adventitia. All right? And almost double the media. So it's a muscular vein. Okay? And a muscular artery is here. Is that clear for everyone? All right, let's move. Okay. So here we have some small muscular arteries. So here, small muscular veins. All right, very thin. Here we have, these are very small, but they have some muscle fibers, V1 and V2, vein 1 and 2, and they're going to drain into vein 3 that is equipped with more muscle fibers. So this is a small muscular artery, and this is a small muscular artery. All right? Okay, now we go to, so here we have this, we go from these arteries, small muscular arteries, all right? And we go to here. And these are also small muscular arteries, all right? And what we have is that these are going to become, become these arteries here. And these arteries are what? 1 and 2. This is very, maybe here there is probably 3 layers of smooth muscle, all right? At the most. All right, here this is even smaller. And then we have this one, which has the same amount of smooth muscle fibers here, maybe 2 or 3. All right, so what do we call this?
Arterial.
Arterioles, exactly, arterioles. So this is an arterial, this is an arterial, this is an arterial. All right, this is the same size as this one. And I bet you that what happens here, so there is almost a continuity here. So this artery is making a kink, all right, an angle. And so here is cut in transverse section, but it's becoming, it's going to change and go, it's not going straight. And so we're cutting these arteries in the same artery in longitudinal section. All right, that's what happens here, just so that we know. All right, so here we have PCV, these 1 and 2, and these are what? C. C stands for
Capillaries.
capillary, right? They're very small. Look, they just are a little bit larger than the diameter of this erythrocyte. So these are capillary. All right, we can tell by the erythrocyte, okay, that they are very small. Okay, so here we have more erythrocytes. So they are larger lumen. All right, so what are these veins? Here it's labeled too. Come on, what is it?
The
I gave it to
post-capillary venule.
Post-capillary venule. All right. Okay. What do we know about? So capillary venule, capillaries, or do they have anything here? They don't have adventitia, right? They don't have even smooth muscle. They don't. They only have endothelium, and it's accompanied by perhaps this could be a pericyte. All right, and maybe this is a cytoplasm of another pericyte. All right, and here is a cytoplasm. Here is a cytoplasm. Oh, and this is a nucleus of an endothelial cell. Okay, so here, what do we have? Do we have in the post-capillary venule? Do we have muscles? No. We have a pericyte too. All right, we have more pericytes, and there is some. Sometimes some authors say that there is probably 1 or 2 smooth muscle cells. Others say that don't. But definitely, they do not have adventitia, and they are aided for their support by pericytes too. Okay, did I say everything here? Yeah. Okay, so this is now I want you to, I need help because I'm losing my voice. This is a arteriole, only one. This is the eosinophilic thing, only one muscle fiber. All right, then we have a few more here, maybe 2 or 3. This is also an arteriole. And this, what about here? Arterioles. And this is, oh, look, another one, arteriole. There's a longitudinal section. All right. So what do we have here? This is a
Capillary.
This is a capillary. There's nothing here. All right. This is this cell here. It's an endothelial cell. This is a capillary, another capillary cut in longitudinal section. All right. So this one now, what's happening is that we are losing the wall, and that's why we look. It looks like we have a lot of erythrocytes. But if you look here, it's probably 1, and here 1. Okay. So, and this is a cell that is found outside of these endothelial cells, right? This one, all right? So this one and this one, not labeled, what could they possibly be?
Pericytes.
Pericytes. So the capillaries are only supported by pericytes. Okay, and so here we have, right here we have, these are endothelial cells. All right. So here are 2, a junction of this endothelial cell with this one. All right, but sometimes there is only one. Okay, let's see what's happening here. Okay, here, this is a junction right here of an endothelial cell, and here could be another junction or could be an artifact. This could be only one endothelial cell. All right, so they're very, very small. But here we have one, and this is one endothelial cell making a capillary. All right, okay, and then we have the fenestrated, the capillaries, and this is a basement membrane. Of course, is outside the endothelial cells. Okay, so we have the endothelial cells, the pericytes, and of course the basement membrane. All right, here are, this is a discontinuum endothelium. There's nothing here, no diaphragm, no fenestre. All right, what's here now? What is this a section for of?
Lymph node.
It's
A lymph
lymphoid tissue.
node.
What? Say again, I did not. Okay.
Lymph node.
Yes. How can you say that? Why can you say that? What makes you think?
It's filled with lymphocytes.
Okay, lymphocytes, yes. But what is the what is the cytoarchitecture?
You could
Alright,
tell
so
the
this
difference
is
between
what?
cortex and
What is
medullary.
what is this? This is a capsule, right? This is a subcapsular sinus. So this is a lymph nodule. Lymph nodule. And this is the cortex. All right. Oh, I even left it here. Look at this. All right. And so, and here is this portion here. This portion here is the paracortex.
Paracortex. Yeah.
Yes, very good. The paracortex. And what's here? This
The medulla.
is medulla. All right. And what's this right here? A trabecula. All right. Okay. So let's for a moment draw the lymph. Lymph comes from afferent lymphatic vessels. Afferent. They are run in they're found in, it's going to occupy the subcapsular sinus, but then runs along the trabecula. And so here is the trabecular sinus, and they are going to get into the medulla. And then the filtered lymph is going to exit with a efferent lymphatic vessel. All right, so here is subcapsular sinus. This is N stands for lymph nodule. All right, so this is what's called this light area right here. Germinal center. All right, germinal center. Germinal centers, here we don't see them. Here, we don't see them. But germinal center occur when, and there are a lot of them, when there is infection, correct? Because there is a stimulation of the lymphoblast to proliferate, and so we are having germinal center, okay? And so what type of lymphocytes are in the lymph node? B lymphocytes. What about in the paracortex? T lymphocytes, okay? All right, what's here? What is a section? Is this different than this section?
I think that is the.
Yes.
It is different?
What is it though? Spleen?
I don't think so. Because this here is this here.
Oh, it's just different magnification.
Yes. All right. So, and in fact, this is light as it is here. This is a portion of a thymic lobule. Thymic lobule. And this is the cortex of the lobule. This is the medulla of the lobule. If something is given to you, you have to look in the light area of these structures here. These are the cassals corpuscles. All right? And here are the cassals corpuscle. And they have only one function. When we look attentively, we see them and immediately we say that we are in the thymus. All right? So they are a sign that we are in the thymus. That's the only really function that is known so far. Okay, so here is thymic cortex, and this is thymic medulla. What's happening in the thymic cortex? T lymphocytes are differentiate into from naive to mature lymphocytes. All right? Okay. So what is the difference here and here? Or is this the same thing? We just discussed that, both. So we have this capsule right here, but this capsule here also. Or this is germinal centers. Is this a germinal center? No. This is medulla. All this is medulla. That is going to get beneath, which is found beneath and extend into the thymic lobule. All right? Okay. What's this from? A section of, what's this? Right here. This.
The spleen.
This is a section of the spleen. All right? Because, who said that? Can you tell me why?
It has white and red pulp.
Exactly. So red pulp, white pulp. And the white pulp always contains this, what's called this? Central artery or central arteriole, to be more precise, because they are small arteries, arterioles, all right? And so this is a splenic nodule. This is red. What happens here and what happens here? Okay, so this is a nodule. Sometimes they have germinal centers. And what happens here in the red pulp? What is the main
Recycles the blood cells.
I'm sorry, say it again.
It recycles the red blood cells.
Yes, exactly. The red blood cells are going to be checked out to see if before they get into circulation, they're first released in the sinuses, which are here. All right, these are the sinuses. It's a high magnification of this, or say this one. All right, so we go here. All right, so this is the venous sinus of the spleen. All right, and these are red cells that are already in the sinus. They are ready to leave the spleen. Whereas these red blood cells that are here, all right? These have to be monitored. They have to be checked that if they are old or they have something that does not agree with the sentinel cells, the macrophages and neutrophils that are in the red pulp and around the venous sinuses, sinusoids, all right? If they are not good, they are not going to enter the venous sinusoid. All right? And so for that reason, there are all these reticular fibers, all right, that are in the wall, and they are in between endothelial cells, and on the outside of these reticular fibers are located all the macrophages and neutrophils and eosinophils that are involved in checking if the blood vessels are good enough to enter the venous sinus. Once they enter the venous sinus, are fine. If they are old, they're going to be recycled, correct, as somebody said. And so we are going to generate hemoglobin, all right, that is going to be put in circulation. Well, it's going to be basically picked up and used for synthesis of hemoglobins. Okay, so here, of a synthesis of, red blood cells. This is a sinusoid right here, all right? These are the red blood cells, the erythrocytes. Here is a schematic view. Reticular fibers, look at all these cells here, all right? This is a process even inside the sinus. And so whatever is not good of the red blood cells, they're not going to get in, all right? So these are going to be checked, and this is actually blood vessels that brings the erythrocytes and all the other blood vessels, they're the blood cells, but the blood vessels are going to be monitored, all right? And here, look, this is a macrophage, this is a neutrophil, all right? And this is a lymphocyte, okay. And so this is, okay, this is the same. Is this the same as this one? Yes, this here, the difference is there is a germinal center, all right? And here, just so that we make sure that we understand and we are not confused, this is a thymus. Is that clear? But this is the spleen. All right, what's this? This is a tonsil, germinal center. In the tonsil, there are a lot of germinal centers. And we know why, because in the mouth, there's a lot going in, all right? And so everything that is extraneous, not wanted, recognized by or sensed by the cells in the nodule are going to be destroyed. So that is why we have this general sensor. There is a sense that there is something not going on. And we introduce a lot of things in the mouth. The food, who knows if it is really clean. It's definitely not sterilized. All right, completely. Okay, so what is this? What is this? What do you think? All right, so let me show you one thing. So here, we don't have villi, and this tissue is going to basically protrude almost and covered by simple columnar epithelium in the intestine. I gave you everything. What is this? Help me. What could this be?
Byers patch?
Yes, Byers patch. And this is the Byers patch in our intestine in our ileum, there are a lot. All right, so this is almost a continuous patch with a lot of germinal centers. Okay, so what also do we know about the Peyer's patch? All right, there are some cells there, right? The monitor, M cells or microfolds that are located in this epithelium that actually looks into all this. Is an extension of this here, which is the intestinal cavity. All right. Oh, I finished. My gosh. Okay, we have time. Are there questions?
Yeah. What was the function of the sinusoid again?
The function of the sinusoid is to collect the blood that contains only the blood, to collect the blood. Let's go back. Okay, collect
So is it the
blood.
same as the venous sinus?
Yeah, it's the same. Sorry. Yeah, it's the same.
Okay, thank you.
Okay. All right. So venous sinus or sinusoid. And so the blood is going, and I showed you here. So here is the blood. And in fact, this blood here has gone through the germinal center first. Because, do you want to know a little bit more? So you have a little bit of the view, the entire view. So here is the artery. Splenic artery comes here and it gives rise to the arteriole, which is in the nodule. This is a splenic nodule. So we have this artery, which has this sheath, right? And the sheath is of lymphatic tissue, all right? There are B and T lymphocytes, all right? Once the artery, which is this one here, yeah, here there are actually already branched arteries, probably, or maybe there are two. Okay, here is one. So here is the central artery, and this is called what? This is lymphatic tissue, and it's surrounded by this lymphatic periarterial, periarterial lymphatic sheath, all right? Because this is the sheath. From the sheath, we have, from the sheath that is around the arteriole, we have there are branches. And these branches come from, precisely from this arteriole, right here. On that, and all right, and then they're going to give, these branches are going to end into these sinuses. Here we have now the sinuses, they're going to get into the red pulp. Actually, they're going to get into the red pulp, leave the blood there, and here are the sinuses, okay? And this is called the penicillary artery. All right, I don't know if that was said in the lecture. All right, and so here we have, this is the penicillar artery because of this further sheath that it has. All right, so from here we have blood released in the red pulp. All right, and from the red pulp is going to be eventually monitored to get into the sinusoids or venous sinuses. All right, so here is the periarterial lymphatic sheath. All right, this is the nodule. This is the marginal zone, and this is the mantle zone. All right, this is a germinal center, And here we have now the artery that exits, and it's going to have this other sheath right here, and it's going to get into the red pulp right here, which is this. And these are the sinuses, and the sinuses are going to contain only erythrocytes that are allowed to go in circulation, meaning that they are good, they're not defective, they're not old. All right, and okay, other questions? Let's see, we have a little bit of time, and so should we go retrogradely? Questions here? Here? This is the same thing, right? The same organ. Okay, what happens here? Okay, what happens here? Maturation of T lymphocytes. And once they're mature, they're going to get into the medulla and go out, okay, in a lymphatic vessel. Okay, here, what do we have? Here we have now lymph that has to be filtered in the lymph nodule, as opposed to the thymus, where the function is to give us mature T lymphocytes. All right? Okay, and let's go back. Okay, so we talked about this. All right, you don't have to worry about this. I don't think that you have to worry about this electromicrographs. But the reason why they're here is that we understand what's going on. Okay, and we know the function. All right? Okay, so here is a pericyte. All right, and here is cytoplasm of a pericyte. And so therefore this is A, and this is a capillary. All right, and here, what do we have? What is this? What is this?
A
Pericyte.
And this is
Endocyte, endothelium.
endothelial cell. And what is this?
Parasite.
Pericyte. Yes, it's not just not labeled. All right. And okay, so these are arteriole. These are all arteriole. All right. And arteriole and arteriole. All right. Any question here?
Dr. Muscala,
Yes.
in the bottom left, are those too big to be called arterioles?
Which ones?
In the
Here?
bottom left, yeah.
Yeah, here. These are small muscular arteries. There is 1, 2, 3 nucleus here. There is another nucleus of a smooth muscle cell. All right, so these are small muscular arteries.
Thank you.
All right, and okay, let's go back. So what is this?
Muscular artery.
Yes, and this is a small muscular artery. This is a medium or medium muscular artery.
Dr. Michelle,
Yes,
the bottom right a vein or an artery? Bottom right.
this one,
It has valves. No,
this one here, this one here.
it's
These are smooth muscle cells.
the one labeled A to the left of that.
I'm sorry. Labeled A here.
Nope, the
Here?
other A on the bottom.
This one here.
The one in the middle on the bottom. I'm
Here.
sorry.
Oh yeah, okay.
Is
Oh,
that-
this one. Sorry. Stop. Okay. Sorry. sorry. Yeah, I couldn't see even the labeled. Okay. Now, so here we have very small muscular arteries, and in fact, it's hard to tell whether they are postcapillary. judging by the lumen here. All right, so, and there is quite a bit of tissue here. But they are very small muscular arteries and they are going to drain, and this is a small muscular artery. And what is this one? Right here. This is really small. And it looks like, this looks like a smooth muscle cell. That is an arteriole. All right, a question on this? Okay. Okay, so.
Dr. Mascala?
Yes.
If you go back to the previous slide, isn't it a vein because aren't there valves in it?
What?
The bottom middle.
I'm sorry, did I say muscular artery?
Yeah.
Sorry.
No, that's okay. I just wanted to clarify.
No, sorry, vein.
Thank you.
What I meant. I'm always have this muscular artery, sorry. Okay, this is a small vein. This is very small veins. Okay. Yeah, thank you, of course. Okay, and so here, these are right here, or these are small muscular arteries, these arterioles, all right? And these are the 1, 2, 3, this and this one, 1 and 2, sorry, 1 and 2, postcapillary venules. All right, and these are the capillaries. And so postcapillary venules, all right. Oh, do they have a tissue? Yes.
For the bottom left image, if we have a choice to choose between arteriole and a muscular artery, which one should we be choosing? Because I was under the impression that muscular artery, we can see the internal elastic lamina.
I did not say muscular artery. I said these are arterioles. It's something between almost becoming small muscular arteries. Small muscular arteries. All right, it's something in between. To have a small muscular artery, we should have at least somewhere between 8 and 10. And here is a little bit less than that. So they're going to be small muscular arterioles. All right. So
thank you.
All right. So okay, what else? Let's move. So here, what is this?
Vein.
Because? And what size of vein? What size of vein? What size vein? Medium. It's a small vein, it's a large vein.
Medium.
It's a medium-sized vein. And how can you tell there is a vein?
The adventitia is double the media.
Exactly. And here?
The media is double the adventitia.
And therefore?
Muscular artery.
Muscular artery. And here? Muscular artery too. It's very thick. Okay, so we go this way. Okay, and what's here? What's here? What is this? It's not the best.
Muscular artery.
Muscular artery. All right, and this is
a vein.
This is the accompanying vein, all right, medium-sized vein. And here is a nerve. This is actually, by the way, a neurovascular bundle, but you don't need it to. It's just for your own information. All right. So here, what do we have? What is this right here?
Inner elastic membrane.
This one here is the internal elastic membrane. And so this is from here to here is the tunica intima.
Intima.
Tunica intima of this particular artery. All right. And this is the
External elastic membrane.
external elastic lamina. All right. It's typical of the muscular artery. All right. Okay. And so here, this is the same. So here is
It's a vein.
exactly, it's a medium-sized vein. Okay. And here?
A large elastic artery like the aorta.
Right. Okay. And so this is to show you how we go from the aorta to a muscular artery. So we go we have to pass by the iliac artery, which shows that it has a sum of and a good amount, as we go more peripherally, a good amount of muscle cells. Okay. And here we talked about that, and we talked about this. All right, here, caveole. We touched on the a little bit, so we have a general idea of how the smooth muscle is contracts. And what do we have here? What is okay? So what is the common cell in all these sections?
Smooth muscle cells.
Smooth muscle cells. All right. Okay. And here we have. Oh, Descartes. Right here, this is a valve, but somewhere here we have the atria, right? Oh, by the way, somewhere here in this region is the sinoatrial node, and what does it do?
Starts atrial contraction.
Exactly. All right. And then we have the bundle of keys. And the bundle of keys, we don't see it because this is actually no, this is the mitral valve, and here is we don't see because it's really requires a section to see a high magnification. But this is the mitral valve, therefore this is the the left ventricle cavity, all right, right here. And so in this ventricle, so here's the valve, and somewhere here, we would have the left bundle of keys, all right, which is made from by the Purkinje fibers put together. Is that clear? All right, and so here are the Purkinje fibers. All right, so the bundle of keys is going to give, it originates from the sinoatrial node because that's how it's contracted. And then the wave, the stimulus, is going to travel via the bundle of keys, which is made by the Purkinje cells. And somehow it's going to be related to all the muscle fibers of the heart. But we are not going to get into that, but just so you see a little bit of the big picture. And so these are, what is this place? here, occupied by, yes, Purkinje fibers. And what is occupied by?
Glycogen.
Glycogen. And so in this regular fiber, or regular fiber, yeah, that's how we call cardiomyocyte. What is around is there is also a light area around the nucleus. What is that occupied by? By glycogen. Okay. Now the other thing is there are also the atria in the atria in the heart. All right, and so there are specialized cells in the ventricle, but there are also specialized cells in the atria. I don't know if I don't have a picture of the atria, unfortunately. All right, but the atria have specialized cells that are regulators of the amount of urine has to be released. All right, so they are. It's called the natriuretic peptides. All right, so natriuretic peptides are secreted both by the atria and also from the brain. And so that helps with regulation of diuresis. Okay, let's see what we have here now. All right. And so what is this? Right here.
An intercalated disc.
Okay, and this here too. Okay, so what is this? Oh, it's labeled. All right, fascia decadens, okay. And there are a lot of mitochondria too in the heart. Okay, and so these are granules of glycogen. Let's see. All right, same. And this is. So what is this right here? How do we call this?
The A band.
Now, what do we call from here to here?
Muscle fiber.
Muscle fiber. From here to here. All right, muscle fiber. And how do we call this here?
Myofibril.
Myofibril. All right. And what is this here?
Myosin.
Myosin filament. All right. And what is this right here?
T-tubule.
T-tubule and terminal cisterna 1, terminal cisterna 2. And what is this right here?
Actin.
All right. And what does actin do during contraction?
It walks along myosin.
Exactly. It slides along. Okay. And so what is this? From here to here. Here to here. Here to here.
Fiber.
Fiber. Fiber. All right. Okay. What is this one here?
Myosin.
No, from the whole thing. From here.
Myofibril.
Myofibril. All right. So it's from here to here because you have to look around. This is sarcoplasmic reticulum that is in between the myofibrils, right? Okay. What else? And of course, what is the beginning of this condition here.
Muscular dystrophy.
It's muscular dystrophy, but
Duchenne's.
it occurs because of
non-functional dystrophin protein.
exactly the functional dystrophy, and so there is the generation of myofibrils, attempt regeneration, and etc. And it's very early, and the individual die in their at the most. Those who are lucky at the age of 30. Okay, so here we have the
Reticulocytes.
and these are
Ribosomes.
ribosomes. Okay, great. All right. And what could this possibly be?
Platelet.
Yeah, platelets. Okay. Where do they come from?
The megakaryocytes.
Yes, that's right. Okay, let's go. This is a
Monocyte.
It's a monocyte. With this monocyte, it's going to become what?
Macrophage.
A macrophage when it's in connected. All these white blood cells are going to to perform their job in connective tissue, right? They circulate, but they get out of the circulation. In fact, what segment in this movement of white blood cells in the connective tissue is going to it's going to allow passage from blood into connective tissue? Capillary, veins, venous, postcapillary venous. Which one of these is the one? This is very interesting. I find it very interesting. How do they go? They move from the blood into the connective tissue. And what do they pass through? What type of vessel that I just mentioned go through? The postcapillary venule. All right, okay. And so let's see what else we have. Sorry about that. Okay, so these are all white blood cells now, right? Okay. And there is a condition. It's just, I think that, I don't know if we talked in the lab, but there is a condition. What is it called when there is a lower number of these white blood cells?
Leukemia.
Neutropenia. All right, it's the number of these white blood cells that makes that condition, that results in that condition. All right. So if we want to increase, what would be a factor that allows the increase of these cells? Granular, monocyte. What about GSF? Granular, monocyte,
Stimulating factor.
colony, okay, exactly, colony stimulating factor. All right, okay. Okay, so, and all right, so everything's clear here. This is a lymphocyte, okay. And here. So we would expect to have a lot of reticulocytes in here. So these are labeled erythrocytes, but there must be Here, for example, right here, this is something that is bluish. That could be a reticulocyte. Okay, so reticulocytes increase, and the increase in anemia. All right, and all right, what else? So here, do you have questions on all this? Do you have questions on all this? No? Okay. So as always, if you have question for me, email me. All right. So I hope that this helps. But email me. I will be checking the email over this weekend between today and tomorrow. Okay. All right. Thank you so much for your attention.
Thank you so much.
Thank you.
Thank you.
Thank you.
Thank you, Dr. Michelle.
Thank you.